Hello, Arthur.
Hello, how are you?
All right, doing well. Um, I don't know who's going to show up here today because Jesse, I think, might be sick,
Oh,
and I don't know if some other people are going to show up. So, how have you been doing, like last week
yeah,
or so?
yeah, I'm fine. I was working on that document that I sent on the Brain Organoid chat. I've made it a lot of progress. I finished it, and now I am moving to the second part, where you try to summarize the knowledge and try to interconnect everything that we learned on the Brain Organoid chat. So I want to start with, oh, what is brain organoid, and we can expect in the future of this technology, how we discovered that it was a nice innovation, and I was reading all the papers and everything that in the PDF that was sent in the chat, and I saw that we have a lot of things to put. Uh, so I will need to spend some time doing this, but it's not a problem. I'm very excited because I think that we can summarize and interconnect each part of that Slack group, because we have so much stuff and so much knowledge, and yeah, I'm very excited, and I will start to do this on Monday, the second part, but I don't know when I will finish, because I really want to make a powerful document, like
Right.
uh, yeah, um, I want to make like a book. So uh, I will make chapters with specific topics, and yeah, it will be like interconnection between an article and a book, and because I will try to write some parts and to put some research and some papers, but at the same time I will follow a timeline, and I think it's this. Uh, yeah. So in this week, um, I finished the first step, and now I'm moving to the second. And yeah,
That sounds good. Yeah, I'm glad you're finding something that you're really grabbing onto. That's good. Um, we usually would like to let people explore and then figure out what they like best on their own. So this is good that you found something. Um,
yeah. It's funny because I had never heard about brain organoids after I started this to collaborate in the order. So when I started to read papers that Morgan sent and that you sent, I started to think, oh my gosh, this is awesome, and I, I fell in love with this technology, because uh, I didn't have so much knowledge, and now I think that I've learned so much, and I'm always trying to improve myself to learn more and more and more about this, and yeah.
yeah. So let's see. That's good. Why don't we go? I have the document open. I can share my screen. We can go over it a little bit.
Yeah.
I liked it. Yeah, I was looking at it earlier. So this is the document here. People are
Yeah.
on YouTube. You can catch up on this. The document is in the Slack channel and the Brain Organoids channel. So if you want to look at it, just open that up. So this is the so these are the different categories. So you have brain modeling, components of the brain, topics without a link, topics on general neuroscience, brain organoids, Evolution, general biology. Okay, that's good. And then these are just the categories, and then these are the references here. You know, have you ever worked on what they call an annotated bibliography?
No.
Okay, so this is kind of what it reminds me of here, where, you know, you have it's a document that people use, and often it's used to guide research. So basically what you'd have is you'd have like these references here, and then you'd work out a short description underneath. So these are good links, you know, these are permanent links, so people can go back later and look and investigate the links. But then you can provide a short description underneath, like maybe 2 or 3 sentences about what each reference is about. So you already have the categories. Brain modeling here, components of the brain, which might also help if we could put in like an image of the brain and different parts of the brain, or describe where these parts of the brain are. But that's something that some people will know and some people won't. So I think some of these are good because they focus on a wide range of things in the brain, like the hippocampus. The regulators is some sort of regulatory. A human tissue screen identifies a regulator of endoplasmic reticulum secretion, I think is what it says. So that's good. That's something that you could kind of provide a couple sentences, or whoever provide a couple sentences of what that is about. And because people, if you look at the title, it isn't immediately apparent to someone maybe who isn't a specialist in that area what it is. So we want to provide the resource, we want to provide the title, and then maybe a description underneath. And we could do this for all these categories. It's fine. We can keep building these categories too. So some of these topics without a link, we might have, actually one area we might have is about induced pluripotent cells. And that's a whole section, a whole area that's very, very large and very hard to get through. So that's something that we would probably want to kind of figure out what references would go in that. It's, people use, I think I mentioned in the Slack, and I'll go over that in a little bit. You can use induced pluripotent cells to create organoids, for example. Or you can use them to create a disease model. So those are, and then we could even provide a little discussion about what induced pluripotent cells are, but it might actually be a little bit better just to have some references to give people on what they are, what are induced pluripotent cells. So, this is nice. So you have components of the brain, topics without a link, and this is again this might go into organoids. Where actually, I'm thinking when I'm looking at this, we have a lot of general neuroscience would be large. That's fine. Brain organoids is pretty large, and that's kind of the...
Yeah.
So, that could, we could have subsections in there, so this one here, how to grow cerebral organoids from human pluripotent stem cells. That's a nice sort of direct connection to organoids. And then, or to IPS cells, as they call them. Or actually, they don't have to be induced. I guess they can be just stem cells, although I think people use IPS cells. So that's a good differentiation point. If we talk about, how do you derive organoids? How do you start? That might be a good place to put some of these references, on IPS cells and stem cells. And then, oh, was that?
No.
Oh, okay. So then, then there might be some more general references like the Organoid Cell Atlas, which would give you an overview of what you're going to be doing. People want to sort of understand organoids. They have to understand it in some sort of sequence, I think. It's first maybe the big picture, and that might be, the organoid cell atlas would be good for that. And then you might have, to start out with, maybe some input, like something in input cells. So what do you put into how do you start the culture? You start it with pluripotent cells, I think. You start with iPS cells or stem cells, and there's some references on that. Then you have maybe some things on protocols. So this is a protocol citation here, and there are probably other ways that or other references here that we might, like making mini organoids, might fit into that category. So it would be like a category of how do you start, and then there other subsections like what kinds of cells or what kinds of things do we want to make. So we our focus of the channel is brain organoids, but people actually make organoids of a lot of different things. So they make liver organoids, and they make gut, and they make all sorts of other tissues, and that's all in the we can understand more about brain organoids if we get information about other types of organoids. So this is all also very important. And then there seems to be this theme in here of like creating structures. So we have sliced human cortical organoids for modeling cortical layer formation. Okay. Model systems for human biology and medicine. Those all fit into maybe this category of, what can you do with organoids, or maybe the previous category. And then sort of like that. And then the last category would be, I think probably like kind of what you can study with it. So this one here, organoids have opened avenues into investigating numerous diseases, but how well do they mimic the real thing? So that's another issue. Like it's also an issue in induced pluripotent cells, where you have, how realistic is it as a model? Because iPS cells are used as a model often for stem cells. They're easier to derive and maintain, but they have a lot of shortcomings with respect to like stem cells. They're not really stem cells. They're not necessarily as stable. We don't understand that stability of their state. So they can go down a cancer pathway, or they can retain features of the cell that they derive from. So there are a lot of things there that are, we really don't understand, and they're not stem cells exactly. They're pluripotent cells that look like stem cells. They have a lot of the markers, but they also have a lot of markers of other things. So, those are nice references to have where, they ask the question, this is a model. People will say, this is a model system. How realistic is it? What kinds of things can you study? So here, organoids is a new model to study neural tube defects. That's a nice reference for, say, like understanding the kinds of things you can study with it. Or inhibiting, Ortho inhibits neural senescence in human brain organoids. This is the action of something inhibiting some process. And so then, this is something, again, like, this is what you can do with these organoids. Exploring landscapes of brain morphogenesis with organoids. Those would all be like in that last category, where you'd have what you can do with it. So that's good. And then I like evolution general biology because, this is like, again, it kind of goes from there, and then it kind of goes more broadly and says, now and we could argue this could come before, but I think it's good after, because once you then talk about like some of the ways you can, what you can do with them, then kind of expanding out to biology. So what is the biological significance of these things? So here we have dissecting Neanderthal brain organoids. You can use it to model a Neanderthal brain. You can look at, cell-cell communication, and we have roadmaps for different things. We have, so this can be organized. But this is my thinking on this. Just treating this as like there is something called a annotated bibliography. So we can do this where we just have these citations. We probably put the full citation in a formal format, but that's later. And then having like a little description underneath where, you kind of talk about the reference. And then having these subsections. So I like the sections that you have, but we can break that down further. And then that's like that part of the annotated bibliography part. But before that, and this may be your second section that you're working on, we might actually work on talking about, building like a review of some of these areas. So brain modeling, for example, would be, why are these categories important, basically? So if I want to start off in this area, or if I want to find this interesting as a reference, why are these categories important? So again, like, with modeling, we know that we use models in biology. We can use computational models, which are like, computer programs, computer simulations. But we also use model biological systems. So like a mouse or, an organoid. And so why are those important? And that may be a whole new set of references that we need to pull into that to cite it, appropriately. But it gives people some background into some of these, like these categories. Why do these categories exist? Why are we going through it this way? And it's just a nice description of it. But then, of course, you mentioned that there's a second part. So that second part would probably come maybe first in that type of thing, where we talk about why the bibliography is important, and then some of the other things you might want to do in the second part. So that's I think the interconnection part is like connecting these categories together.
Yeah, this first part was just to organize the research that was sent in the chat. So because I saw that the Brain Organoid chat was created in 2019.
Yeah.
So there's a lot of things, and when I started to think on creating a document talking about brain organoids and everything, I just imagined that I needed to create a document like this to organize with everything that was sent, because it will be more easily to me when I will write the new document, the second part. It will more easily to find information within a document with a lot of links and divided by topics,
Yeah, this is great. I like it. How far back did you go on the channel?
Since the beginning. I started in 2019. So
oh, wow. Okay.
This is everything.
All right, good. Yeah, we started this channel. Actually, I started it a long time ago to sort of get started, and then this was before Morgan got involved even. And then Morgan was putting things in, and we were kind of losing track, because it was like we would just put things in there and forget about it. Or, it would like pile up, and you'd have like, and the thing about Slack, or the version we use, is we don't really have, we have a message limit. So sometimes these things will literally get lost. So that's good that we have this.
Yeah.
So, yeah, thank you for your hard work on that. I think it looks good. And I think if other people want to join in on this, I think that would be great. I think maybe, we can just revisit this from time to time and look at the strategy of what we want to do with it. I think that this part is good, having this as a list, and then, augmenting it. And then the second part, whatever you want to do on that, we can review that and put them together in a document. I think, oh, is that?
Um, yeah, for the second part, I think that it will be lovely to have people with me because I will start to write, and I really need to make a document with a lot of information, a lot of text, a lot of images, and a lot of references. So, uh, of course, just me, it will be, I will need to spend a lot of time, and but with more people writing, and it will be more easily, I think. So yeah, I welcome everyone that wants to work on this uh, large paper about brain organoids to join me and send me a message, and I will explain everything on the Brain Organoid chat on Monday. So yeah,
Yeah, that sounds good. Thank you. Yeah, and so we'll be working on this. And then in terms of publishing it, there are different ways we can publish it. We can use something like Wikibooks, which is like we want to be able to edit it over time. I think that would be important. So we might use something like Wikibooks, and I know we haven't talked about how to do that, but we'll talk about it.
yeah,
It basically uses like we could host it uh, on our GitHub, or it's like there's a Gitbooks option. There's also a blog option on GitHub in a GitHub repository. So these are things that are like ways to publish it where we can edit things as we need to. We can issue like formal releases. We could even do this like on something like Figshare, where we have a formal release. We turn it into like a PDF document, and then that would be like version 1, and then version 2, and then it would keep going. And I mean, we could even publish it at some point. I don't exactly know where we would put it, what journal or anything, but we can definitely
yeah.
publish it as a digital document.
I have absolutely no knowledge about published
Yeah, I
things
know.
because I am in the beginning of my undergrad, and I don't have any experience with publishing papers and things like this, so I will need help.
Oh yeah, definitely, definitely. I think yeah, we'll once we get it, get some momentum behind it, we'll have like a lot more. It'll really take shape. So that'll be good.
Yeah, thank you.
You're welcome. So let's see. Next order. Thank you again for that work. And again, if people want to do something like that, that's great. We can, give me or Jesse or someone else a document during the week, and then we can bring it up at the meeting, and then we just go through it. We just try to like give advice and think through what things and again, if you want to do a presentation, I know Jesse put out a call in Slack this week about presenting things. So we're trying to get people to present more this year. This is a good example. You can prepare a document to review in the meeting, or you can create like a 5 to 10 minute thing and update on your ideas for something. Don't feel bad about it if you think it's not good enough, because we want to encourage people to just get started on things. Like, if you have an idea, like I think you've seen in the meetings where I've done this, where I've just put an idea out there or a series of ideas that maybe aren't totally connected, but you can see where, maybe some opportunity there. That's what we want people to do more of. And sometimes this will be like directly related to a project. Sometimes it's not, but we can always figure out how it fits into things. One of the great ideas of some of the greatest ideas kind of come from like reading some paper and saying, oh, this is kind of an interesting aside, and kind of thinking about how it fits into maybe what you're interested in or the greater effort in the scientific literature. So, this is something that it's a skill that you have to build over time, and it's a very useful skill because it gives you the opportunity to find new frontiers and sort of work there and make new discoveries. So it's a very important skill, but you have to start somewhere. So I'm encouraging people to, just send, just kind of come up with something. It doesn't have to be much, but we can cover it in the meeting and we can get rolling with things. So very good. I'm glad that Arthur is doing that, and it looks like Aaron is doing that, and of course Ankit is doing that as well. So it's all very good. So the next order of business is, next order of business, I'm going to share my screen. We talked about, let's see, I'm going to go to the Slack. I want to kind of review a couple things in here. So the first thing is actually this is the thread. So I want to go to Brain Organoids. And so let's review maybe some of the stuff that we've had. This will kind of serve as a project review. So you can see that, again, we have a lot of papers that come in, and and so this is Arthur talking about starting to work on this project. This is the link to it. We have some publications. Why don't we go back to maybe last Sunday, because I think we've talked about maybe some of these things previous to that. I mean, I think last week we talked about this brain wrinkles and folds matter. So this is a paper on what they call purification of the brain, of the human brain. And I just found a paper, and I don't know if I have it to show this week, but that this is something that is found in mammalian brains, and there's sort of an evolutionary common ancestor where we can see this purification, as opposed to the smoother brains you see in mice and in rats, and I think in dogs. You have these gyri, which are these folds. And I mentioned last week that the human brain is a huge sheet, this neocortical layer, and it gets folded up into the skull case. So as the brain, the skull doesn't grow as big as the sheet grows, so the sheet has to be folded in. But this scarification is a trait that can actually be, like, humans have it, other primates have it in a lesser degree because their neocortical volume isn't as large. So, but this folding is something that's actually a trait that has a common ancestry in mammalian brains, and then in the mammalian phylogeny or tree. And then there's another trait in mammalian phylogeny, which is a smoother brain surface, and you find those in other organisms. So it's kind of, I'll have to bring that paper up maybe next week, but that's a very interesting point about what I talked about last week. So this is where we can use brain organoids as models to study development and evolution. I don't think this is the paper, this is something else, but that's a good reference. Morgan has a paper on here on this is a summary of, sometimes it does this in the browser. Okay. Brain 2.0 Transforming Neuroscience. So this is a NIH Brain Initiative summary. So this is a paper where they summarize this project or this initiative in the NIH. And this is the U.S. Funding agency where they fund work on the brain. It's the National Institutes of Health. So the way that's set up is that you have the NIH, which is an umbrella agency, and then you have different sub-agencies underneath. So, like, the NIH has, like, institutes on diseases. They have institutes on infectious diseases. They have institutes on other aspects of health. And so each of those sub-institutes has a director, and then there's a main director. And the main director of the NIH, Francis Collins, is retired, and there's an interim leader now. So Anthony Fauci is actually one of these sub-agencies. He's the head of one of the sub-agencies. But this is the way it's structured. So they're able to, like, because they have that kind of structure as an organization, they're able to make these sort of strategic initiatives across their sub-agencies and even working with the NSF. So, they have to coordinate all this. They're trying to bring all of the sort of, expertise to bear on this one problem. So this is their initiative here where they create, in this case, they're working on sort of understanding the brain to create a brain cell atlas. So this is where you're creating atlases of every cell in the brain, or, slices of brains. So like the Allen Institute, which is a private institution, has like this huge, database where you can go through and you can go and look at like a mouse's brain slice by slice, and they've annotated all the different cell types. They have precision analysis, which is where you can focus on very specific things in the brain, and then connectivity analysis, which actually involves some machine learning to segment out different things in the brain and figure out how the cells are connected together, and then building a database. So this is actually a lot of computational stuff, and NIH doesn't have any one sub institute on computational stuff, but they actually can draw a lot from what's going on with sort of other bioinformatics studies and what the NSF is doing. So this is a nice project here. So this is where they're trying to fund a comprehensive human brain cell atlas. And so there's a lot of things that we can kind of extract out of brain tissue. We can slice the brain tissue up with what they call microtome. You get these very thin slices, and you can look at different things in those slices. You can also break down brain tissue and look at where the cells are lysed or blown apart with chemicals, and you get a lot of biochemistry that you can look at. So people will sometimes take tissue samples, break apart all the lipids in the cell bodies, and then you're left with a lot of biomolecules you can analyze in samples. So you can do a lot of things. Ramoni Kahalu, who was one of the first neuroscientists, developed some staining techniques. So we use a lot of staining, which is where you put stain on different parts of the brain. They may bind to different chemicals, and you can look at these different things in the brain. So, there are a lot of old techniques where people are doing things like staining and looking at slices, and then there are newer techniques where you're using, like genomic data, and you're sort of doing a lot of computer vision to sort of segment out parts of those images and give us a database of things like that. So there's a lot of stuff we can study, and having an atlas where you put all this together is really what they're trying to work towards. And this is a whole brain effort. So, you take structures from all across the brain, you bring them together, and you can build these wiring diagrams. So we've talked about connectomics before. We have organisms like C. elegans and Drosophila and we've seen these in previous meetings, where we have these nice, beautiful maps of connectivity, but we don't know necessarily the function of those maps, and we don't really have good maps for, say, like a human brain or even a mouse brain. It's just kind of like a thing. It's a work in progress. People have made a lot of progress on this. It's just not, we need more data. We need to synthesize more data. And then, once we have the synthesized data, we learn a lesson from the Genome Project, which was also one of the human genome projects is sponsored by the NIH, which is that once we have all this information at a very precise data at the appropriate scale that we want to look at, what do we do with it? And so, the human genome has been out there for 20 years, and it's been refined over that time, but we really still don't know how the genome works. If we want to look at like a complex trait, for example, like some disease state in the brain, or even just why the brain is organized the way it is, We know some of that, but it's really bringing the full power of that database to bear is still kind of a work in progress. So I like this initiative, but just to remind people that once we get once we get these data in place and these databases, there's a whole bunch of work that needs to be done to interpret the data and to put it into a proper context. So that's a nice paper. Thank you, Morgan, for that. So we have some more things like brain assembloids. So assembloids are actually related to organoids. This is the paper here. So why do they have to define assembloids in this paper? I'm not sure I can get my fingers on it here. So they just say brain organoids and assembloids generated from human pluripotent stem cells can model early developmental stages of human brain development. I assume assembloids are just kind of like collections of cells that they grow up, and they're not really organoids, because organoids require some supporting vasculature and other things. So this is a nice overview of this type of approach. So we have brain organoids, which are these tiny organs that grow from the pluripotent state, which is a stem state, and then they get differentiated through your chemical medium, maybe your surface, different cues that you can deliver to the cells. So this is what they call, they have organoids, and then they have assembloids, which it looks like they're more like just different collections of cells that are put together in the same context. So that's good. So the Assembloid approach described here leverages self-organization to enable cellular crosstalk. So this is where cells are communicating. And for those of you who don't know, cells actually do a lot of communication between each other. So when cells are in the body, they're constantly signaling one another. And if they're in close contact, they can signal each other much more easily than if they're far apart. But in the body, you have like the bloodstream, you even have the nervous system where these signals can diffuse over long distances. But when cells are in proximity, they share a lot of signals with each other. And this explains sometimes why in a lot of organisms, if you take like a stem cell and you put it into a tissue, if you transplant that stem cell into a tissue, it will acquire the fate of the surrounding cells in that tissue. So if you this is what they do in cell therapy. They take stem cells, they take cells that aren't committed to any one fate, they put them into a tissue, and they differentiate to some new state, and the state of the cells around it. So this is something it doesn't happen by magic, it's this intercellular signaling. So this is the power of assembloids. We can use that feature of a lot of animal models, like mouse and human. That's the way it usually works. It doesn't work in some organisms, like C. elegans, because those cells have a programmed fate. So, but in any case, this assembloid approach is sort of maybe a happy medium between deriving some sort of organoid and having a cell culture where you just have cells on a monolayer on a dish, or a single layer on the bottom of a dish. And so this is the Assembloid approach described here leverages self-organization to enable cellular crosstalk, including through cue-mediated cell migration and directed axon path finding, and allows advanced functional probing and manipulation of the resulting neural circuits. So in assembloids, the component parts can be virally labeled with human cell type specific reporter viruses, and these are these labels that allow us to see the cells and their different features of the cells under a microscope. We can use reporters such as fluorescent markers, and we can see these things. This is why people are doing this type of thing. But you can study these things in assembloids. That allows you to study cell autonomous effects in disease models and probing neuronal activity of specific cell types as circuits self-assemble. So these cells here will form, say, like a neural network in a dish, or in, wherever they're growing these assembloids. The signaling happens between the cells. They maybe send out axons to, to communicate with one another, to make connections, and then those things can be studied, outside of the context of the brain or outside of the context of some biological tissue. That's really kind of a stripped down version of this organoids approach. So this is good. This is a nice paper going over assembloids and other types of things. So thank you again, Morgan, for that. We have some papers on applications to neural development. We have some papers on this technique, spin infinity. This is a bioreactor design, so this is how they grow a lot of organoids in this bioreactor. This is a chamber for growing things and controlling the environment. So this is, Morgan has been doing a lot of work on kind of figuring out how to build a biological lab to do this in, and figuring out how much it costs and everything. So he's trying to get a lot of this information. And so maybe in our document we can address some of this. I know Morgan has a lot of work on it, but it could even be a Medium article, even with the cost focus. So we might do that as a Medium article, and then have this larger document as a way to kind of, like say you want to get started in organoids, we can, give you some information about how, a lot of the things that you need to build a lab, and then, maybe, references so you can understand what kind of approaches you need to take. And so we can mix like Medium posts, which you've seen that we have our Medium, with these more formal documents. I think that's a good way to go on this project, because it's I think there's a lot of stuff that is rapidly changing. So a lot of this stuff with the hardware and scaffolds, that state of the art is growing pretty fast. It's like, probably next year we'll have to update it because, there's so much progress being made. So that's another concern for like, sort of formalizing a document is what is maybe going to last for years. Like a lot of these references that Arthur's put in the list, those are things that are foundational. It's not going to change a lot. They're going to be new references, but but something like this where we have protocols and hardware, those things change a lot because we get better hardware, we get different protocols. So that's good that we're kind of working. I'd like to hear from Morgan at some point about this, some of what he's found out. I think I commented here where we're talking about, just kind of, how do we deal with a lot of these issues like, cell culture lab space and the microscopy and all the things you might need. It's very, there's a lot of planning to even do the science here. So, so this is about using microfluidic devices to look at, to sort of mimic a extracellular matrix. So extracellular matrices are, in tissues, that's one of the things that you miss in cell culture is that matrix in between the cells. So there's a lot of supportive, biochemistry in that matrix. There is a lot of physical interaction with the matrix. So these are things that we miss in cell culture. So this is a good paper for understanding that. There are also some nice, this is a nice paper on bioengineering and, some of the issues with that. Nanoscale extracellular vesicles, which are, again, part of this matrix, intracellular matrix. And then, let's see, there's some more things on pricing and other assorted issues. This is a nice review feature of cerebral organoids and drug discovery. A nice review. Then there's a set of papers that Morgan put in here. I can't remember. This one's on cell maturation, hallmarks, triggers, and manipulation. So this is like a paper on kind of sorts of things that we observe in cell maturation. So these are all kind of like how a cell becomes specialized or mature is important. We have our what we call stem cells, which are the cells at the base of differentiation. They can take on a number of fates. You have pluripotent cells, which can differentiate into 4 or 5 different fates. This isn't, what they call totipotency, which is where you take a cell and you can create a whole new organism from that one cell. That's not what this is. Pluripotency is more restrictive, and it's just stem cell being able to create, say, like a neuron or a skin cell or a muscle cell, just depending on the genetics of that cell lineage or the signals that it's receiving from its environment. So, but our mature cells then are the things that are differentiated. And by and large, they can't go backwards, although in some cases they can. There's famous cases of like regeneration and, even moving to a cancer fate is considered sort of going back more towards a stem cell state. So this kind of goes over like the hallmarks of maturity comprise changes in anatomy, form, genes, circuitry, and interconnectivity, and physiology. So, and then those confirm adaptive behavior. We discuss efforts to harness their chemical and physical triggers in vitro and in vivo. Now, maturation strategies may support disease research. So this is all about like these different signals, and this kind of relates to what we talk about when we talk about switching in our lab discussions. So that's a nice paper. Morgan put together a collection of papers in here, and I'm not going to do it justice because I don't really know. It's another review. Okay, so this paper here is the first paper. This is on electrophysiological measures from human Ipsc, which are these induced pluripotent stem cells, derived neurons. So you can derive neurons from these pluripotent stem cells. These are associated with schizophrenia, clinical status, and predict individual cognitive performance. So this is a nice paper on schizophrenia, which is a heritable neurodevelopmental disorder. And the idea is you can study the heritable nature of schizophrenia using induced pluripotent stem cells. So you could theoretically take a cell, like a skin cell from an individual. You can do some sort of personal genomics on that individual, on that cell. And then you can convert that cell or its cell line into a induced pluripotent cell. And then you can study, because it has the same genome, it's just changing the signals, telling it how to behave as a cell fate. And then you can actually study these disorders. So you can actually look at, in that cell, you can understand some of the things that are going on. You can differentiate it back into a neuron to understand more about the disease. So you can look at things like electrophysiological measures, and you can look at different features in the patient, because now you have a link between the cell line and the patient, and they have the same genome. So this is a nice paper on this, and I've seen people attempt stuff like this before. It's a nice model because you go from like individual differences that you would see in individuals. You can go from like cognition, and you can go down to the cellular level, and you have these links that allow you to make predictions about the disease. Now, you can't do this with every disease. It has to be something that's highly heritable. And we might say that, like, if you start changing the fate of cells, that's it loses sort of its cognitive context or neuroscientific context. So, there are pros and cons of this type of approach. Then this paper should be paired with this one, which is human brain organoid networks. And so this one is kind of talking about sort of how do we understand neural circuits in terms of creating organoids. So they created organoids and they've created these networks between the organoid cells. So the organoid starts with like a pluripotent cell. You get differentiated neurons, and then you get these networks that form between the neurons. And so we don't really know much about the physiological behavior of these neural circuits in organoids. So this is something, actually, when we started this project, I was working with Aidan Rock, who is doing other things now, but we were talking about modeling connectivity between cells in an organoid. And we were talking about how you would do that, how you would look at the origins of the connectivity, because these cells are going to form these circuits in the organoid. And it's much easier to study this in an organoid than it is in a developing brain. So they use these high-density CMOS microelectrode arrays, which are these arrays that have a lot of sensors in parallel, and then and also shank electrodes, and then record from the organoid at different time points. You can capture local field potentials and single unit activity. So you can actually look at the type of fields that are created in a brain, in a tissue, that have or implications for cognition, but also single unit activity and how that's synchronized as this circuit is wiring up. And so you can actually look at all these things. It's very, very robust data, I think, coming out of a method like this. For spiking activity, we estimated a directed functional connectivity graph of synchronous neural network activity, which showed a large number of weak functional connections and meshed within a network skeleton of significantly fewer strong connections. So it has this sort of, in connectomics, they call it the rich club effect, where you have some cells that are highly connected and have a lot of sort of organizational power over the network, over the circuit, and then cells that don't, cells that are on the periphery. So this is an interesting set of findings. We demonstrate that human brain organoids have self-organized neuronal assemblies of sufficient size, cellular orientation, and functional connectivity to co-activate and generate field potentials. So this is a really cool paper. Again, it's functional neuroscience in organoids. And then Arthur had a question about where we start with cells, and I gave a good answer here. Basically talking about pluripotent cells being the origin point a lot of times for these organoids. And then, you can push the stem cells or the induced pluripotent cells around with transcription factors or media conditions. So this is, we talked about this in a previous lab meeting when we were talking about this initiative to kind of create a human embryo out of a single reprogrammed cell from an individual. I might donate a cell or a cell line, and then it's reprogrammed into a stem cell, and then it's used to create an embryo. I can't remember the intermediate steps there, but they've worked it out. They're trying to work on this problem, and we had a discussion about this. It was somewhat skeptical. So if you go back to that conversation and you see, I'm not sold on the idea. Let's just put it that way. So, and then this paper here also is the third paper of that, or I guess this is related to this explanation I gave. So this is something Morgan provided. Actually, maybe this is from the first thread that I showed. This is transcriptional programs regulating neuronal differentiation are disrupted in knockout stem cells. So there's a knockout of this gene DLG2, which just means they inactivate that gene or they remove it from the genome. So it's not expressed. And so then they're able to identify this gene and this knockout as sort of a causal factor or a risk factor for schizophrenia or some related disorder. So again, schizophrenia has a very strong heritability. That means that there are genes that are involved that we can look at that will control the expression of this phenotype later in life. or maybe in adolescence. But the idea being is that they have this genetic background, and the idea is that if in organoids you can knock out these genes. Sometimes you have to knock out certain alleles, but this is just basically something that you can't do in a behaving human. You'd have to get a induced pluripotent cell, knock out the gene, and then grow an organoid, and then you can observe the behavior, the electrical behavior of the cells in this organoid, and then it can maybe correlate with what you see in a schizophrenia patient. And so they kind of go through a lot of this. There's a lot of genome-wide association studies that are drawn from here, so we have an idea of what the genetic basis is. And again, this isn't possible for every trait, but in schizophrenia you can do this. So that's an update, I think, of that project. Did you have any questions about that, Arthur? I know that's the project you're in right now.
No, I think that is fine. It's a good thing.
Yeah, I just wanted to provide some context for a lot of the things we're throwing into the channel, and make sure people are aware of what's going on. I think it's very robust, the project. And again, I think it'll benefit from some of these documents.
Yeah, sure.
Yeah. So let's see. That's that review, and let me go to the next thing, which is. So I've got a couple things I want to close here before we move on. Jesse, I think, was talking about some things for organizing information, and I know we had talked about like some things last week. I know he talked about Obsidian and Markdown, so I'm not really sure where this is. It could be in our admin lab channel, which is private, and I might look here. Actually, let me look up here. Oh yeah, okay. So Jesse talks about this thing called Obsidian, and it's a tool. It's in Markdown. It allows you to do things with visualization. It allows you to do things in the language of Markdown, which is it's not HTML, and it's not text. It's sort of a way to write documents, and you can do this in GitHub. Write formatted documents with headings and hyperlinks and all sorts of things. It's not HTML, but it renders kind of like an HTML document. But it's good, useful for like writing documents where you want to do a lot of hyperlinking, and you want to have like code embedded in it and everything else. So Obsidian is this tool that you use. You can use it with Git. You can use it with Markdown, and it's a nice tool. I don't know too much about it other than that. Jesse is not here today. He's not feeling well, so maybe we'll review this next week. And this actually came out of, this is a nice tool, but this is actually coming out of a longer conversation about different tools that we might use for different things in the community, or in the lab. So we talked about using like GitHub for organizing contributions, for scheduling different deadlines. We use other tools, and then this Obsidian would be nice for like generating documents and contributions. And I wrote a post, and this is something for another group I work with here, and this is a blog post I did almost a year ago now. This is something I'm going to call the full stack community. And so what that means is that we have, in an open source community, which we kind of are, not truly open source software community, but we do a lot of these things. We draw a lot of inspiration from that. And so we talk about a full stack solution. We mean like we have a number of things we want to accomplish, and we have all these technology tools to do that. So we have a stack of like maybe 6 things that we want to do, or we have a number of things we want to do. We have a stack of 6 technologies that we sort of rely on, and we want to integrate between them. So if someone comes into our community, into our lab, we say this is the way we want to do things. You join our GitHub, you join our Slack. We have our meetings in a certain platform. We have certain things we want to do for publishing things. they're all like standardized. We don't like, switch between 3 or 4 different tools for the same task because it's hard to invest. Like you have to get to know these tools, and it's hard to spend that much time learning 4 different tools, and then having people do it in one tool versus another, and then having one tool go out of business, so we have to convert to another tool, and it gets really unwieldy. So there has to be some sort of organizational principle here. What I've come up with in my other work is that there's some sort of full stack open source solution for this. So what I mean by that is that there are a lot of things we want to do. We want to create docs. We want to create spreadsheets. We want to meet virtually. We want to take notes. We want to deploy software. We want to have events like hackathons or lab meetings. We want to have like repos for things like papers or code. We want to host a wiki. We want to have media that we distribute. And then we want to have things like playlists, which we can archive information on. So actually, I say Rockwire Community here, and this is what we do there. But we also, in our lab, we do a similar thing. And this is something that it's sort of a overview of it, like a sort of almost like a theory of this sort of approach. I know Jesse's been working on this, but I think it's good to like look at it from this vantage point, because you can see that there's like this intentional strategy of different tasks that you want to do, and then you map them to different technologies. So for docs, we have Gdoc or Atom.io. I've talked to Ankit about Atom.io. It's very good for creating documents in LaTeX, or if you want to create like something in Markdown, but you want to convert it to a PDF file. This is a nice tool for that. We also have Gdocs where we work on different docs, like one I just showed earlier in the meeting, and being able to share them openly and having the tools be free or near free, because we don't have, or we have a large budget for things. Tools can become very expensive. And quite frankly, sometimes tools will lock you into a certain mode. So like with Slack, Slack is a nice tool, but when you get to a certain number of people, you start sending out a lot of messages. And then when you send out a lot of messages, there's a limit on the number of messages Slack allows from the free version. And so if you have like 50 members to your group and you start posting messages and you start to reach your limit, then you can either do one or two things. You can subscribe to Slack, which is expensive for 50 people, or you can move to a new platform, which means that you have to either take all your old messages and move them over, or abandon what you were doing entirely, that ecosystem, and then move to something new. And so neither of those solutions are good. And I know we chose Slack early on, and I like Slack, but that's just a limitation of what we have. So this strategy might allow us, to kind of think through, and a lot of these tools are not like, they don't last forever. Some of them are actually quite fleeting. Some of them are, we don't know how they're going to be used as we scale up. So it's kind of hard to know these relationships in advance. But I think if we think through it like this, where we have these different functions, and then we have these tools, at least if we have a diagram where we know what we want to do, and then we know what the tools are, and then we evaluate the tools and pick the best one. I think that's a good strategy. And then you can assemble a stack of tools. And so this goes to this onboarding issue that we've been talking about, which I'll move to next, which is if someone joins the community, and I know Arthur, say, is a recent person in the community, what do they need to know to do certain things? So now Arthur is using Gdocs for creating a document. That's good. We can share the document, shared editing. But like if we go like to Ankit, who's working on something that's more specialized, does he know how to use Atom.io? I talked to him and he said he has some familiarity with it. But this is the kind of thing, we want to have the stack of things. So if I want to go from a meeting to creating a document, I go to like my Jitsi space, and then I can, maybe go to this Atom.io to do something. And ideally, at some point, maybe there are integrations we can work on. So like, taking notes in Notion, we show that as a screen share in Jitsi. We maybe go from Notion to some document that we want to work on. And then from there, we might publish it to some media. So the media is WordPress and Twitter. It's just like, we want to create tweets. Do we want to advertise what we're doing? Do we want to write a blog post? Do we want to write like more, substantial document that we want to archive? That might be in this playlist category. I should have also put like, formal publications or formal releases of publications. But that's the idea here behind this this full stack idea, is that we have this stack of technologies for different functions in the community that allows people to learn like a subset of things. They don't have to learn everything. It doesn't become overwhelming. There's what we might call a tool zoo, which is where for any one thing there are like 10 tools possible. And we might use different tools. And it's not good for collaboration if I'm using one tool and you're using another. So getting everyone on the same page there. And then if we go between functions, which we often do, we can actually find ways to integrate between these things. So that's what I meant by this full stack community. Now, this onboarding guide, which we're working on, is kind of like, the idea is to take that full stack community and just kind of lay out the different things that we do in the community. This is a presentation I did that's an overview of the lab. This is very high level. But what we want to do is actually create an onboarding guide, which is more specific, gives us this stack of technologies, this stack of functions in the community, and then how do people want to approach these. And so that's what we're going to do here. And we're going to provide it to new people to bring them into the community. And that should be good. I think it's a good way to, like, it can be bewildering, I think, to come into a new environment and kind of figure out how to do what. Well, how are we going to do this? So I think that, and then we may also do something for like, create categories like programming, or, how do I get to presenting an idea even? Because this is a stumbling block, I think, for a lot of people. So we'll work on that as well. And it fits into that same model. So, any questions on that, Arthur?
No, completely fine.
Oh, good. Well, I hope people find it useful. And we're going to be working on this onboarding guide over the next several weeks. And we are putting out a Gsoc project, so that's going to be good for that as well. People coming in and saying, I want to work on this Gsoc project, and how do I get started? That's going to be very useful for people like that.
Yeah, it's a pity because I really love Slack.
Yeah, well,
I will miss Slack.
I don't think we're moving off Slack. I'm just saying that, like, one of the drawbacks is basically we're going to lose old messages, and there isn't a good way to archive it. So,
yeah, good.
But yeah. So yeah, and then I guess we'll move on to the Gsoc thing. So last week I talked about Gsoc and our potential proposals, and it didn't look like we were very far along on that. But I did get a project from Inkit, and he suggested a project, and I haven't finished that yet. I might go through that and try to work out the description a little bit. But this is the one that we worked out for AI ethics. And so I think I just showed like a very rough outline of it last time. But this is actually what's been submitted to INCF. And they liked this project. They said it was very interesting. So we'll see who we can get for this. So this is called a collective cognition model for AI ethics. And this is really kind of based on what I think some of what Aaron has been talking about, and some of the things we've been talking about. I think Krishna is also like kind of the inspiration for this, some of Krishna's work. And so this is going to bring someone who can do, who can build models. First of all, we want them to build a sort of a systems model, and then we also want them to build an agent-based model. So what we want to do is we want to be able to evaluate data, real-time data. And, this is important in a number of socio-technical contexts. So this is where, people might take real-time data, like images from cameras or something, and they kind of put it into a database, and then they analyze it using machine learning. Or, it could be just some sort of system where you're doing monitoring of a complex socio-technical context, like trying to predict, economic variables or something. Now, this isn't actually what this is about. It's a little bit different. But our group is looking to develop an auditing system and a simulation of collective cognition that will improve open source community sustainability. So this is very specific to open source communities. So we have a lot of interactions in an open source community, and we actually can get data from GitHub repositories. I'm not sure if people large organizations, large open source organizations publish this data. I'm not entirely sure about that, but this is something that exists. It's definitely measured by GitHub. What I mean by that is if we have an open source project, we'll have different things that are done in the community. We'll have pull requests, we'll have issues, we'll have follow-up issues. All those things can be measured. If you want to look at a repository and how active it is, you can look at how many pull requests have been made, how many modifications to some, how many software releases have been made, how many issues have been filed, these sorts of things. And ideally it would be a graph over time where you have some, I can use my board actually for this. So ideally, your data would be some sort of set of interactions over time. So this is a time series here, and then this is some variable, and then you have this fluctuation over time of interactions. So this might be the number of issues filed in a repository, or the number of pull requests made. You can see that there are fluctuations. The thing we want to avoid, and this is kind of where this is going, is that you have this time series where involvement just goes down. All right. And then that's okay. Maybe no one's updating the software. So we might have another part to this graph where we look at software updates, or versioning or whatever. And we might get little spike or little lines here where we get this and this and this. So you can see it's being updated, but it's not being maintained in the same way. So, you start off early with a high level of maintenance and it goes down. Or we just kind of stop updating it altogether, which is also bad. So what we want to avoid is a situation where we don't have people sticking around to modify the software as it needs to be. Because there was in the ethics channel, there's an article on, there was a thing in the US on, it was a White House task force or something. Basically, it was this Log4j virus, and it was because Log4j is involved in open source maintenance. So the idea is that there's this virus, or there's a security bug that's prevalent in certain open source software, and it needs to be fixed, but sometimes some projects don't have the staff, they don't have the ability to fix it. So some of these open source tools that we work with or rely on can't be repaired quickly because there's no one there to say plug a hole. If you had a piece of software from Microsoft even, that may also be an issue. But at least there's some corporate strategy. In open source, we typically don't have that strategy. So this is Google calls for new government action to protect open source software projects. It's basically that we want to coordinate with the government to help open source organizations to make these critical bug fixes, because people oftentimes will use open source software. Linux is open source. It's not closed source like Microsoft. So if people are using Linux operating systems, And in many countries, they do use that as the predominant operating system for their government agencies. I think Brazil does, and other countries. So if they have this security breach, it has to be the open source community has to be the place where that solution originates. And with something like Linux, it's pretty easy because it's a pretty robust community, and so it does get fixed. But the problem is in some communities, maybe in community where there's less robust, open source sort of sustainable community, you get this graph on the left where there's this loss of participation and sort of response time to these issues. So that's what we want to avoid, and that's why we're doing this project. We want to kind of sort of put some sort of thing together where we can simulate the behaviors in open source communities, but also create a systems solution where we can look at how these things are self-regulated. So if you're successfully applying to this program, you'll be creating a homeostatic system, which encourages cooperative and altruistic interactions, but also use simulated data generated through an agent-based model of these type of interactions. So you'd create an agent-based model of an open source community, you'd run it, you'd get some data, and then you can benchmark your system, which is like it has feedbacks. It has that cybernetic quality where it has feedbacks. You have variables, and you put data in, and you account for the feedbacks. You account for the feed forwards. You account for other things, and then you can audit those relationships and see where they go. And so this will help maybe build algorithms later that we can do to sort of encourage things, virtuous behaviors in an open source community, or a way for people in these smaller organizations to predict what needs to be done, how to prioritize things, and maybe to encourage participants to take on different roles. So that's the idea here. Some of this, like I said here, you can in the case of creating a cybernetic model, you can create these traditional box and arrows models, which are the ones that I've shown, I think, before in meetings, where you have these different boxes which represent variables. So you have X, you have Y, you have feedback from Y to X, you have an output of Y, and it might go to Z, or it might go to the outside of the system, and then Z goes to the outside of the system, but Z actually also has a feedback to X. This is the input. So you can see that there are these complex relationships between multiple variables. There's complex causality, which we talked about last week, and then there's also just this kind of structure where we can define the variables as things going on in the open source community. You have outputs, you have feedbacks, you have inputs, and all those things can be quantified and then used to design an algorithm. And then defining these variables is actually quite important for the agent-based model too, because the agent-based model will use those variables as agents, or the properties of the agents. So this is going to be kind of hard. I think this is going to be a longer project, and hopefully, we don't expect you to create the perfect solution over the course of the summer. We just want you to get started and move forward on it. So you can use these box and arrows models. You can use something like reinforcement learning as well, which is just, with agents that choose policies, and that would fit more directly to an agent-based model. Or, you can take inspiration from Mick Ashby's Ethical Regulator, which is something we've talked about in past meetings. I've talked a lot about this with Rob Stone, who's a collaborator who doesn't attend the meetings, but he's got some interesting things to say about cybernetics, and he's been interested in this Ethical Regulator. So Mick Ashby is it could be the nephew of Ross Ashby, who was the very famous cybernetics person from the 50s and 60s and 70s. And he's worked on this thing called the Ethical Regulator, which is basically how to create good ethical situations through regulation, through cybernetics. And so this might inform, this idea, and there's some papers here, might inform your model for this part of the project. And then there's this other model, which is sort of based on cultural evolution, which is agent-based. And it's, again, taking what you've done in the first model and applying it to building these agents. And so I don't think I mentioned how to build it. Okay, maybe down here. Okay. I suggested that people use NetLogo, which is a nice platform. I think it uses, I think, Java for building the models, but I think you can build the models in a number of languages. They have stock models, which is why I suggest this. They have models that are already constructed. You just need to modify them. So that's an advantage using NetLogo. The first model, though, the cybernetic or homeostatic model, can be coded in Python or Julia. So these are, scientific computing languages that you can use for this. There are other options, but those are the ones we kind of want to focus on, so that people can use it and plug in data, and or you can use Kotlin as well. I didn't mention that here, but so that's the two-pronged approach here, and I think that'll be useful. Like, again, I don't think you need to really produce a really strong result by the end, but you need to work on like these two parts, and get to a reasonable point where we can like continue it next year, or maybe we can do small scale experiments with it. So that's the goal here, and I think this will be one of the longer projects. I know they have the link, they have like the short project and the long project this year. I think this will be a longer project. So if you're interested, contact me, or you can even join our Slack and talk about it there. So that'll be an interesting. We'll see who we can get for that, if we start to be able to recruit people for this. I might start posting it on social media pretty soon, because I just sent it to the INCF group, and they're excited, but it's not quite the application period yet. So we need to get some buzz for it to get people's attention. Hopefully we get some interesting applicants. The way I usually approach it is I usually will give them this kind of problem, pretty open-ended problem, and say, can you solve this problem? Pick an approach. Given what I've told you about these things, how do you propose to solve this? And so, usually that's a good way to get people to do some really innovative stuff. They'll go through the application period and they'll create a model in some language, and they'll demo it at one of our meetings. In the DevoWorm group, that's how it's done. And then by the time we have the application period, we have like about 10 applicants who have really nice work, really good work. It's hard to pick one person for that project because there's so much good work. But then, it also gives you a nice pool of talent to draw from. And people can, if you don't get selected, you can continue your work in the lab. That's what Jesse did with his project in 2019. He joined he applied to this project, and he put in an application, and then I said, do you want to join our lab? And then he did, and he really kind of found his place, I think. So it's always a good thing to get involved with things like that, because you never know where they'll lead. So good.
Yeah. Bye, Bradley. See you. I needed to go now.
Okay, that's fine. Well, thanks for attending.
Thank you so much,
All right.
and I will go today to the Brain Organoid chat with information regarding the next steps of the document and what I will do on Monday. So thank you so much.
All right, thank you. Talk to you later.
See you.
See you. Bye. Okay, so now I think we'll finish up the meeting, the last 30 minutes, with some papers. So no one's in the physical meeting right now, but you'll see this on YouTube. So we have, let's see, we've gone through a lot of these things. Let me close some of the windows down. I did want to talk about a couple things here. I don't think I need to share my screen. I can just do this. So the first one is this Locomotion Vault. And this is something that was published as, I think it was I found this online. I thought it was really nice to look at. This is a human-computer interaction workshop output. So this is the CHI Conference on Human Factors and Computing Systems. And this is a paper, or I guess it's a paper, Locomotion Vault, the extra mile in analyzing VR locomotion techniques. So this is a nice 10-page, or it's a 10-page paper, but this is like sort of like their interactive simulation that they put online. So this is from, I think it's from a lot of data sets that exist on different things you can do in VR. So the idea is that you put on your headset, you put on your haptic gloves, and you can interact with things in the environment, and you can instrument people quite a bit here. So one of the things to do is to put people in, put some VR goggles on, put some haptic gloves on, touch gloves, and maybe some other things, and then you can measure their interactions within the virtual environment. So you can simulate parts of your body that don't exist. You can look at people's skin conductance, which measures their arousal in these environments. You can have them simulate freely walking around a room. You can have them simulate walking into nature. So as we've talked about before, there are a lot of ways to look at different ways to interact with the environment. You can look at haptic feedback, which means you can touch things, virtual objects, get some sort of touch feedback, and it stimulates things in your brain that you would get if you did the real thing, hopefully. But what's nice about virtual environments is you can set up all these different environments pretty cheaply, and you can measure not only interactions, like movement, like you can track movement of arms and heads and hands, but you can also track physiological states. So you can measure brain activity, you can measure skin conductance, which is an indicator of emotional arousal. You can do other types you can measure muscle activity and other things. So there's potentially a lot of things you can measure. And so people interact with these environments. Now, this is where you have the similarity of different things as people are interacting. So some of these measures are going to align with one another and some don't. And so this is what the similarity measure is. So the idea is some of these environments have similar human responses. So as you can see, you have these different environments here up in the corner. And shooting games, manipulating objects, navigation. You have this, walking on a surface that's moving. There are a lot of different, doing these, like this is rowing in a canoe. So there are a lot of different activities, and they have similarities in terms of their expert similarity and database similarity. So database similarity is a similarity of these databases is classified by sort of their content. So you can look at what's involved in this. Rowing, walking on a treadmill, some sort of action where you're moving your arms and legs. And then there are other things like navigation, where you don't map it directly to what you're doing in terms of movement. And then this one where you're doing, there's haptic feedback. You're walking through an environment, but it's passive until you touch something. So there's that. Then there's expert similarity, which is, what's the human response to these things, and how similar is it? So this is a nice data set, or this is a nice display because it kind of analyzes a lot of these different data sets. So this is this is a data set where you can enter a new technique. You can, suggest changes, and this gives you information about the publications that these data come from. So there's the year where things are cited. So let's see if we have gamepad. There are different ways we can look at these data here. You can look at some of these things for a gamepad and then motion direction, where there are different types of motion. So there's gaze, marker, location, walking, vehicle. So there are different things that these studies look at, and you can get a sense of so in these, so device I think is informative, fixed. So there are different ways we can look at this. Controller tracking is actually quite common, where the investigators are tracking the controller, and that gives us some information about some of these environments. This is a nice database for that. So it's called Locomotion Vault. It's about people moving around in VR and analyzing these locomotion techniques. That's a nice little tool. So then I want to talk about some more, let's see, I'm going to go to papers. So, let's see. First thing I want to talk about is this, first thing I want to talk about for that is this moral machines. I know I've been talking with Angela, or Oidis as she's going by now, about some of the work she wants to do as a PhD student. And so one of the things she's talking about, she posted on life logging, and I think it was the Ethics Society and Tech channel. She's looking for volunteers to do some life logging to get some data, and she's trying to connect it to this idea of moral machines or effective morals. And so I want to go over a little bit of that just to give people an idea of what the citation space is there. So there's a group at MIT a couple years ago, they published this paper called the Moral Machine Experiment. And this is, so the abstract here reads, with the rapid development of artificial intelligence has come concerns about how machines will make moral decisions, and the major challenge of quantifying societal expectations about the ethical principles that should guide machine behavior. So this is about decision making in machines, but ethical decision making, as opposed to just making decisions based on, some criterion that's not really linked to their moral context. This is a problem with, not just with pattern recognition, but with the machine understanding whether what it's doing is right or wrong. Should the machine fire a gun at a crowd of people, or should it not do that? I mean, that's a very absurdio ad reductum example, but it's still something. I mean, with self-driving cars, for example, the self-driving car sometimes hits pedestrians, and so if we can have it understand that, pedestrians are not to be run over, if there's some moral framework that needs to be put into place, then that's something we need to think about. So to address these challenges, we deployed the Moral Machine, an online experimental platform designed to explore the moral dilemmas faced by autonomous vehicles. Now, this is actually about autonomous vehicles, so that example I gave is very good. This platform gathered 40 million decisions in 10 languages for millions of people in 233 countries and territories. So this is where this life logging comes into play. So we have a lot of data here, basically, and they're trying to, so they describe the results of their experiment. So they summarize global moral preferences, first of all, and they have to kind of come up with a basis. And I imagine if Angela uses her life logging data, if it's collected, you have to decide how you want to collect the data and analyze it. That's what I told her. I said, we're basically going from something that's qualitative, which is like people's recollections of what they've done or what they're doing in their daily life, and then creating sort of maybe these variables that I talked about before, where then we can take, a sampling of different things that they're doing and relate them to one another. So in this case, they're summarizing global moral preferences, and I imagine some of that is like looking at similarities, but sometimes if you compare across languages, you don't really get a good comparison. So there is a lot there that we need to look at the paper more deeply to see what they did. So second, we documented individual variations and preferences based on respondents' demographics. Third, we report cross-cultural ethical variation. and uncover 3 major clusters of countries. So they've been able to uncover cross-cultural variation, which might inform what a machine does, because a machine is, not, it doesn't really have a culture. I mean, it could have a culture, but the idea is that in one culture's ethical standards might differ from another culture's ethical standards. So we don't know what that underlying, if there is such a universal set of morals, what those are. So looking at this cross-cultural, we get a broader sampling of what that might be. And so they come up with 3 major clusters of countries, which is encouraging because it suggests that there's some underlying structure to moral decision making, and we can maybe exploit that. Fourth, we show that these differences correlate with modern institutions and deep cultural traits. We discuss how these preferences can contribute to developing global, socially accepted principles for machine ethics. All data used in this article are publicly available, so we can actually draw from these data sets. I don't think that there's, the thing about these, I think this is a Nature paper. The thing about Nature papers is that they don't have a lot of, the articles are rather short, but the supplemental materials are rather voluminous. So this is something that we're going to have to look into more. So they kind of go through their techniques here. These are their sampling points. So they sampled across the world, different cultural contexts, every continent except Antarctica. And then they're able to kind of analyze all those data and kind of classify different ethical rules. So there's a German example where there's this, consider as a case in point the ethical rules proposed in 2017 by the German Ethics Commission on automated and connected driving. This report represents the first and only attempt so far to provide official guidelines for ethical choices of autonomous vehicles. So this is important context for interpreting the findings. And maybe other countries can follow this German example. German ethical rule number 7 unambiguously states that in dilemma situations, the protection of human life should enjoy top priority over the protection of other animal life. So the autonomous vehicle, I guess, can hit a squirrel or a deer, but shouldn't hit a human. If the choice is between a human and a deer, then the choice is to not hit the human. In this figure, you see this. This is almost like the Trolley Experiment. I know people probably are familiar with the Trolley Experiment because it's become very famous. But it's like you have to choose trains going down the tracks. And you have to choose whether to go off the tracks or hit people on the tracks. It's some variation on that. And so in this case, it's whether to hit these pedestrians or hit this barrier. And so if you're just using some sort of, computer vision technique, you might identify the barrier and you might identify the humans, and the car might have trouble distinguishing where it should go. It doesn't want to hit the barrier because it's an obstacle, so it might just plow through these humans because it doesn't think human life is much more important than hitting this barrier. You're going to crash either way, but maybe it's better to hit the barrier. I mean, this is just one of these examples. The data just operating on like standardized, segmented image data might tell you that, avoid all obstacles. But if you have a choice between hitting one obstacle and the other, to make the distinction and to make the appropriate decision. So this has a lot of, I think, relevance to what Angela's doing, at least in terms of thinking about how to analyze the life logging data. So they have a number of different global preferences here, sparing different individuals in different situations, sparing different types of humans, sparing the law. You can violate the law if it means that you can spare a human life, because an autonomous vehicle wouldn't necessarily know that. It would just either follow the law, would follow the law to the letter, and it would maybe sacrifice some pedestrian in a crosswalk. So these are things that the machine has to understand. And so then they have these cultural clusters, which are, there's a Western cluster, which is North America and many European countries. There's an Eastern cluster, which is in Eastern Asia, but also some of the Southern Asian countries and Southeast Asia. And then there's a southern cluster, which is Latin America, which is sort of, I guess, its own cluster. There's some connection to the Western cluster, but not always. So that, and I don't think they mention Africa in here. I think it fits into the southern cluster. But the idea is that you have these different cultural traditions, and there's different different preferences within those clusters. I think that's what they're getting at here. So they're different, preferences for these different rule exceptions. So this kind of gets at where we are with this map here with the different traditions. This is a tree that kind of branches out to the edges here. So these are different countries and where they kind of fit into this classification. And notice they imply that there's a root here. So this is helping us understand the sort of universal set of moral judgments for this sort of thing. So that's one paper. Then there's a social neuro AI paper, which is by a group. They put this. Maybe this is like a conference paper or something, but this actually talks about sort of social neuro approach to AI. Social interaction is the dark matter of AI, as they put it. We are making the case that empirical results from social psychology and social neuroscience, along with the framework of dynamics, can be of inspiration to the development of more intelligent artificial agents. Complex human cognitive architecture owes a large portion of its expressive power to the ability to engage in social and cultural learning. So again, this is another sociocultural inspired computing or robotics paper. People have been doing social robotics for a while, but I think there's this opportunity to integrate cultural diversity into this. And we've talked about this in prior meetings. And even in the poster that we did on our embodied developmental AI stuff, where we have this the one preprint that we haven't generated here is this idea of a dual process or dual inheritance model, where they're inheriting some sort of, the agents are inheriting some sort of innate feature, and then they're inheriting some sort of cultural or behavioral feature. And so the idea is to look at those things as they evolve in the agents, either independently or together. So we might actually do something like that for A-Life this year. We'll see how that goes. But so neuroscientific theories of cognitive architecture, such as the global workspace theory and the attention schema theory, can enhance biological plausibility. Second, intelligence occurs in time as opposed to over time, and this is naturally incorporated by the powerful framework offered by dynamics. Third, social embodiment has been demonstrated to provide social interactions between virtual agents and humans and a more sophisticated array of communicative signals. So there's a focus on communication as signals, this idea that there's this dynamical system that we're looking at instead of just kind of outputs over time. And then third is these cognitive architectures that we look at with different theories. This makes the agents more sort of biologically plausible, or perhaps socioculturally plausible as well. And so that's what they're arguing in this paper. And then this was something that I got for Angela in case she was in the meeting, but this is a nice document on how to choose a research topic. This is actually from the effective thesis. I think this is something she sent me as one of the things that she's looking at as a way to this is a nice paper because it kind of focuses on this effective the kind of community she's looking at, the futurist community, and they actually have some nice guidelines for choosing a research topic, getting interested in different things, and then looking at things like long-termism and some of these other issues. So, we can approach problems from different perspectives. If you're interested in coming up with a research topic or, kind of guiding your project, I think this is a very nice reference. This is, I think, organized for sort of what Angela's doing, but I think you can get some insights out of this if you're doing any kind of project, really. Understanding the impact of social movements. This is like a lot of, solving, like addressing large social problems, but, it's a nice way to kind of think through hard problems. So there's that. Okay, so now I want to go to this set of papers. Actually, this is a nice set of papers for what we were talking about with Arthur. And this is an article on systematic reviews or scoping reviews. So this is guidance for authors in choosing between these different types of reviews. So this is put out, I think, by some library scientists, and they're making the difference between a systematic review and a scoping review. So a review, of course, is where we take a lot of literature that's existing and we put it, we kind of review the state of the literature. We've talked about this with our different trajectories papers, the cognitive trajectories, the epistemological directories, and some of the other things that we're working on in the group. So, but I think these two different types of reviews maybe are enlightening to people who want to get started. And I know Jesse's doing this preprint for his trajectories work. He's kind of putting some flesh on the bones of that. So this might be useful to him. So scoping reviews are a relatively new approach to evidence synthesis. And currently there exists little guidance regarding the decision to choose between a systematic review or scoping review approach when synthesizing evidence. The purpose of this article is to clearly describe the differences in indications between scoping reviews and systematic reviews to provide guidance for when a scoping review is and is not appropriate. So what is a scoping review? So systematic reviews, and this is in healthcare, began to appear in publication in the 70s and 80s. With the emergence of groups such as Cochrane and the Joanna Briggs Institute. In the 90s, reviews have exploded in popularity, both in terms of the number conducted and their uptake to inform policy and practice. So systematic reviews go across a number of fields. You see the field of evidence synthesis is seeing the emergence of scoping reviews. So the systematic reviews are kind of a way to go through the existing literature and kind of see where, different categories of things that have been studied, doing this systematically so you don't just kind of throw references together. You kind of organize it around different topics and kind of make sure that you've sampled the literature evenly and completely. So there are a lot of different techniques to this, and I think this paper gets into some of them. But scoping reviews are different, and this scoping review is where they're similar to systematic reviews in that they follow a structured process. But now they're a valid approach for circumstances where systematic reviews are unable to meet the necessary objectives or requirements of knowledge users. So there now exists clear guidance for scoping reviews, how to conduct them, and the steps involved in the scoping review process. So there's these references 6 and 8. I don't think I can link to them in this document, but so let's see. So indications for systematic reviews. Systematic reviews can be broadly defined as a type of research synthesis that are conducted by review groups with specialized skills, set out to identify and retrieve international evidence that is relevant to a particular question or questions, and to appraise and synthesize the results of this search to inform practice policy, in some cases further research. So systematic reviews actually to focus on a topic. Sometimes you can focus on a field, and so in that field you'd be saying, what does this field look like? And I think in Jesse's example, or in his case, that's the question you're asking. You're asking, what is this? What's the shape of this field that we're looking at? Or maybe sometimes, what are the interdisciplinary connections between fields? Systematic reviews use explicit systematic methods that are selected with a view to minimize bias, thus providing more reliable findings. It follows a structured and a predefined process that requires rigorous methods to ensure that the results are both reliable and meaningful. And so you basically undertake a systematic review to confirm or refute whether or not current practice is based on relevant evidence, to establish a quality of that evidence, and to address any uncertainty in the variation or practice that may be occurring. So it's almost kind of like something like a meta-analysis, where you take different studies and you evaluate the results of those studies, and then you kind of look at the variation over different studies to see if they kind of point in one direction or not. In this case, it's more of a review. So you're going to be reviewing a number of different subcategories of papers. You're going to be bringing that evidence to bear and seeing if, maybe what that means for the structure of the field. And then, where do we go from here? Do we, can we construct these trajectories that Jesse's asked about, or can we do other things? Does it lead to new research questions? And then the scoping reviews are an ideal tool to determine the scope or coverage of a body of literature on a given topic and give clear indication of the volume of literature. So this is actually what I was talking about with respect to a review, like what we might do on a topical review. So this is a scoping review where you're examining emerging evidence, where it's still unclear what other more specific questions can be posed and valuably addressed by a more precise systematic review. So this is maybe kind of what Arthur is proposing, which is where you have this set of topics that we want to know something about them, we want to put them in some context, and see where what the frontier of this field is. So this is the difference between these two types of review. I wanted to bring this paper up because I think it's useful for thinking about some of the things we're doing in the lab. Like Jesse's approach is maybe fundamentally different from what Arthur's proposing, but we need to know kind of how to, where that fits into these different types of reviews, so that maybe if we go to publish one of these, we can follow a methodology that's a little bit more focused on what's expected. So this kind of lays us out in detail. If we want to kind of follow up on this to kind of get maybe a sense of how to do these reviews and how to do them better, that's a good resource for that. I also then wanted to talk about this Robert Rosen paper. So I'll finish up with this, and then that'll be the end of the meeting. So Robert Rosen is a person who he's sort of a theoretical biologist. So he's done a lot of work with looking at biology and physiology very theoretically. So he's interested in looking at biological systems, in this case a physiological system, using mathematical models. This is in the International Journal of General Systems. And this is, I don't even know if this is published anymore, but this is something from 1986. Robert Rosen published a number of books. He's actually done a lot of interesting work with like category theory and some of the other tools of, not really artificial intelligence, or, well, I guess like we talked about Michael Erbeeb's work in category theory for robots. So this is actually kind of related to that. This is looking at causal structures in brains and machines. So he's got these things called MR systems, which are basically a system to look at different types of systems where you have different components and their relationships, looking at function and regulation. So why don't we get into the paper? It'll become more clear. So this is, I think Robert Rosen's dead now, so I don't think that this is, you can find out more, but he has a very large body of work. So if you're interested, look up some of his books. Maybe we talk about him as well in the DevoWorm group, so you might go back to some of the videos of those meetings. We present some formal and empirical evidence to support the proposition that maximally constrained mechanical systems exhibit some of the basic properties of organisms and of machines. We show further that although such maximally constrained systems seem thereby extremely limited in their possible behaviors, they can be programmed to introduce a limitless plasticity in their behavior without breaking their constraints. We then turn to the ambiguities inherent in the word machine and show that programming in the above sense introduces a new relation among machines. namely simulation of one machine by another. The simulation relation has none of the nice properties familiar from mathematical machine theory, basically because the causal structure in the simulator is completely different from the corresponding structure which is being simulated. So this is an interesting paper. He's done a lot of work on looking at these MR systems as relational systems, which is related to category theory and some other areas. So the idea is you're relating one set to another set. So it's like in category theory, you're using I think a functor to move from one category to another and map things from one set to another. Or it's not a set, but it's a category. And he's done a lot of interesting work. He's shown, for example, that I think there are a couple of different debates really deep in the literature about some of the implications for the Turing Church thesis and other things. So he's coming up with this idea. He's starting in biology and he's kind of applying this to artificial life and machines and machine intelligence. And what's interesting is that he's doing this from the standpoint of biology, whereas most people do this from the standpoint of like the brain or some sort of behavioral sources. So it's kind of interesting. He starts in like different biological systems and moves towards machines instead of maybe the other way around, or at least ignoring some of what the organism is doing. So no one knows or can express what an organism is. The best we can do is try to say what an organism is like. For at least the past 400 years, the pervasive metaphor in biology has been that of the machine. But no one can say what a machine is either. So, what are the definitions of these things? And it kind of starts in that sort of philosophical way because, we're making this set of relations in organisms. So organisms as a metaphor have been likened to chemical factories, telephone exchanges, or more recently computers. But these are all metaphors. And so, can we design like a relational system that's maybe unique to organisms, or at least kind of breaks down those metaphors a little bit more in more detail? So we want to sort of clarify how organisms are like a machine, or like a chemical factory, or like anything else. And, this actually has implications for considering what is life like and what is not. So there's some other debates on, whether what Rosen is saying relates to sort of the definition of life. And so it's really interesting work. Let's see if he gets into what he gets into in this paper. So he goes through the history of some of this. He goes through some of the ideas of the relationship between organisms and physical constraints and things, what brains are doing as well. And so, machines do the same type of things as biological systems? And so to really kind of make this very abstract, you can look at two different ways. You can look at this in two different ways. So purely on physical grounds, there are only two classes of material systems which exhibit this kind of hysteresis, which is where if you deform a system, it doesn't return to its original state. So if you pull on a rubber band, the rubber band will snap back. But sometimes you pull on a rubber band, it deforms the rubber band. So it's a lot, it doesn't return to its original state. The deformation is called hysteresis. And so hysteresis is an important part of this equation. So on the one hand, you have essentially random systems like fluids, where the velocities of specific molecules are unimportant, and only the averages count. On the other hand, there are constrained systems, as we shall see. Such systems are precisely those for which velocities are determined by configuration. So if organisms are not random in the above sense, they must be then strongly constrained, and it is those constraints which are responsible for their behaviors. So this is this constrained systems approach, where you have organisms that are constrained by physics, and then it constrains their behaviors in space, and it leads to these sort of maybe quasi-intentional behaviors. So constraints for T form the essential ingredients in its distinction between laws and rules, and between the alternating layers of dynamical and linguistic behavior, for which he has argued are characteristic of organisms. So this is, again, drawing to these different behaviors in artificial intelligence or in human cognition, which result from this organism and sort of its physical constraints. That's kind of an interesting paper. This is brought to my attention by someone I follow on Twitter. But I've been familiar with Rosen's work, but he has so many different things that he's published. It's kind of interesting to go back and look at some of these things. So let's see if there's anything else in here. We don't really talk about MR systems in this paper. I'm kind of disappointed, but we can talk more about MR systems in future meetings. So I think this just kind of goes through some of this constrained control. Although such a maximally constrained mechanical system it would appear an extremely special kind of system. Although such a system is capable of behaviors which are vastly different from those exhibited by other mechanical systems, driven by impressed forces, they still only have one degree of freedom and are hence essentially special purpose devices. How then can they be likened to such things as bacterial cells or other biological systems, which seem to manifest a limitless plasticity? And by plasticity, it means, they can evolve, they can adapt to their environment, they have multiple functions or behaviors. Cells have a lot of different functions. Special purpose devices are more sort of in the machine realm, but actually viruses are maybe special purpose devices. So we have things that are sort of on that boundary of living and non-living. And so how does that sort of functional description, how does that define life? One answer to this lies in something we've already alluded to above: the transition from an autonomous dynamical system to a control system. So we're looking at like a dynamical system, which just looks at the dynamical state of a system. It could be a mechanical system, it could be a non-living system, it could be a living system, to a control system, which is of course a machine, but also is something that transitioning to something that actually engages in control. And so if we combine that with what we know about the Ethical Regulator theorem, which we've talked about in previous meetings, you see that there's this relationship between control and sort of this good regulation. And it's sort of more than a dynamical system, that it has to have this sort of adaptable nature. And then, of course, then this leads back to what living systems do, which is they're able to predict regulation and control it in a way that a dynamical system does not. So a machine would have a corresponding dynamical system, and a machine might also have a control system, but that control system would be, it wouldn't be regulated from internally, it would be regulated by some governor or some external agent. So this kind of goes through some of this, considering some what's going on here. We can think of the specification of a particular forcing function, which is beta xt, as a program. So this is something that they bring up here, beta xt, which is a function. This is basically talking about the relations locally where you have a memory and it's holding a sufficiently small number of configurations in that memory, or in that, sort of state buffer. So the parameters of beta sub I through which forcings are manifested are in general functions of configuration as well as time. So stated differently, the mapping related parameters to tangent vectors is itself a function of configuration. So basically you're reconfiguring the system. You have a small subset of different trajectories that the system can take based on some of the constraints, based on some of the forcings of physics and inputs to the system. So, you have this control system that's forming, and it's basically constraining the degrees of freedom selectively. It could be in a memory, it could be in just from physical constraints. But this forcing function, this beta xt, can act as a program. So that now we have this idea of having a program, an autonomous program perhaps, that can do these things and can drive this adaptation. So this could be something that you could find in an artificial intelligence system. You could find it in a cell. You could find it in a brain. And I guess the difference is that it's more guided by physical constraints or less guided by physical constraints, or has a different capacity in terms of the number of local or the number of different configurations it can hold. And this idea of configurations is really kind of related to information theory. It's related to thermodynamics, but it's also related to information theory. So the idea is, how many configurations are you holding in memory at once? The more configurations you have, the more information there is, because you have more things that can happen versus fewer things that can happen. So this is a nice, these are nice things that, you can go back 50 years, 60 years and find the answers to some of the questions we're working on today. Thus, these systems are programmable, even though maximally constrained. So they can be very constrained. This forcing function may only hold a few configurations of the system or potential trajectories, but they're still programmable. You can still, choose which one you want. And in fact, having it in a reservoir like that actually improves the ability for the system to control itself, because it's not trying to decide across every configuration which one to take. So this is crucially important to our subsequent discussions on machines, cells, and brains. So considering these things as concrete objects in nature, you have machines, you have cells, and you have brains. Now he gets into mathematical machines, automata, and neural nets. And this kind of gets into some of this idea of having software programs, or maybe things that look like software programs or can be interpreted as them, as the basis for maybe the brain or what cells are doing. So he kind of gets into Turing, he gets into algorithms, he gets into some of these issues. And John von Neumann actually had a nice tool, and people still use this. It's called the self-reproducing automaton. And so this is the idea that you have these programs that can reproduce, and they can develop patterns, and they're little modules of pattern formation, and they execute their program and they form a pattern in the environment, and then they reproduce themselves and they form a similar pattern. They can be mutated, they can reproduce, they can replicate, they can die, they can even have a developmental period. So this is an interesting mathematical tool that exists that is so the idea of a universal computer that Turing argued for, von Neumann said that this mathematical machine implies the existence of a universal constructor. So each of these programs is a universal constructor. It can execute commands, and it can reproduce and execute more commands, and you can have these constructors that exist. And if you ever look this up, self-reproducing automaton, you'll see these maps where they're these little patterns that sort of emerge over time in a tree, and this is just these universal constructors being born and living their lives and constructing things in the environment. And so he thought of the latter apparently as a material device. So this universal constructor is a material device. His argument was that construction, or the execution of a blueprint, was no less an algorithmic process than the computation of a program. And so you can make this connection between the execution of a blueprint and the execution of a program. And by blueprint, he means blueprint like, a genome expressing itself and creating pattern formation. So the creation of an organism from an embryo would be the expression of those genes, which would be the blueprint for. Now, I think a lot of biologists probably think that's a little naive, but no matter, he's making the connection between this kind of blueprint, or even a biological blueprint, or a cognitive or neural blueprint, and a program. So that's, thinking about it in computational terms, that maybe still be a pretty powerful metaphor. And so then we get into simulations and models. We talk about some of these things, and this is where he gets into this mapping and some of these things. And then mathematical machines and material systems, he kind of gets into this in terms of sort of the details of what this might look like in a material system. And then he talks about these mappings quite a bit. So these are the mappings that he's getting into. And I mentioned before. And so then he talks about causality. This is a longer paper, so you might want to look at it in more detail if I'm missing anything here. So then he kind of gets into causality and how that plays a role in all of this. Okay. So I think this is a nice paper. Definitely, some of the ideas you're going to run into are maybe dated. Some of them are novel, and even to like most people working in these areas, it's pretty novel. Some of them are controversial. So we have, different ways of like relating things mathematically and then mapping them to living systems. And then, we're autonomous systems. And, there's a bit of, there's a lot of mathematical formalism here and a lot of like representational content here. So, the idea is basically if you create these metaphors for living systems, you can use this by mapping functions from one to another and demonstrating that process, but also kind of thinking about what kind of process would work best if you had like this set of needs, this set of physical constraints, this set of configurations that a system can take, and then representing that as either a dynamical system or something like some sort of memory system with a memory or system that monitors local states and then can operate on them. So there's a lot of mathematics here, but it really kind of boils down to that. And then some implications, you have things like the self-reproducing automaton, the origin of life problem, which of course is where you have a living system that emerges out of nothing, or maybe like a inert physical system. And then relational models and their realization. So he talks about these MR systems, which are relational cell models. These are metabolic systems where you basically look at these two sets and the mapping between the sets, and everything in these different sets are relational. So one thing relates to another. And so that's in here. And then development and pattern generation, he gets into that as well. So this is something I don't want to get into too much, but Turing developed a model of chemical morphogenesis, and he's putting that in this context. There are things like the Jabotinsky reaction, which is this spontaneous chemical reaction that you can generate in a chemical medium. It forms these scroll waves that are very complex, but it's not driven by any intelligence. It's just the chemical reaction unfolding in the physical constraints of that system. So, in like Turing morphogenesis, it's very similar. You have these chemical gradients the agents respond to and form they form striping patterns or other types of patterns that are like, local systems that respond to global signals or even larger local signals, and they perform these patterns. And so all of this is I think it's very relevant to artificial intelligence because, we're talking about like systems that recognize patterns and form, causal structures. And how do you get to that? What's the origin point of that? So this actually relates quite strongly to our developmental AI stuff. So that's that paper, and I'll post that in the chat. So I think that's it for today. I want to thank Arthur for attending, and hopefully Jesse gets better soon. And I know Ankit is busy today, so he couldn't join us. But if you're interested in joining our group, go to our website, orthogonal-research@weebly.com. There are links to the meetings there. We're also active on Twitter, where sometimes you'll find a link to our Slack, or email me or someone in the lab, and we can get you connected. Have a good week.